first thing that makes a good scientist is curiosity. I always think of the phrase that Alice said, curiouser and curiouser. That's the sort of attitude you need to have. How does it work? Why is the sky blue? It's like being a child. You're always asking questions. And it's that curiosity, that always asking questions, which is really important, I think. That's the key to being a scientist.
Professor Fran Ashcroft studies the machine we rely on most in our lives. If any part of its delicate construction breaks down, we're in real trouble.
The human body is completely amazing. It has so many different control systems. So many things are being regulated. We don't have to think about breathing, we don't have to think about the regulation of our blood pressure, we don't have to think about our temperature control, we don't have to think about how our blood sugar level is controlled. All of these things just happen automatically.
I study physiology, which is how the body works. And the thing I'm particularly interested in is how the blood glucose level, the blood sugar level, is controlled.
Fran's journey to the top of her profession began as an inquisitive child in Dorset. From a very young age, she was fascinated by the world around her.
When I was a child, we were allowed to run through the fields. It was just utter bliss. I was out from morning till night. In those days, there were lanes which had not yet been tarmacked, and I fell through the hedge into one of these and I found a whole host of wild orchids. They were only common spotted orchids, but to me they were the most beautiful things in the world. And it was then, I think, that I became a naturalist. And from a naturalist, I progressed to being a scientist.
But Fran's constant curiosity could sometimes embarrass her teachers.
Sometimes I was a bit of a trouble to my teachers because I was always asking questions. Probably the worst occasion was when I was about 11 years old at my secondary school, and we were being taught about reproduction, and we had done chickens. We had done how birds mate. And then we had gone through both the male and the female sex organs. I understood all of this perfectly, but there was just one question that I couldn't really understand, and that was how the sperm got to the egg. It was obvious in chickens because they put their bottoms together, but it was not obvious to me in humans. So I asked the question, and there was this kind of scandalised silence. And then the teacher said, "Frances, don't be so cheeky." But of course I wasn't being cheeky. That was a problem. I really wanted to know. I had no idea. And the whole class turned round at me and they looked at me and they said, "How can you ask that question?"
Fran realised that once she started asking questions, she couldn't stop until she found the answers.
So what happens to the body at altitude? So why don't penguins' feet get frostbite? How do babies manage to keep warm when they don't shiver? What are the problems that the human body encounters when you go underwater? If I was instantaneously transported to the top of Everest, I would pass out within a few minutes. On the other hand, if I was to walk up there, slowly and gradually taking my time, I could probably manage it without oxygen. Why should there be this difference? The answer, of course, is that the body is able to adjust if it descends gradually. One of the things that happened to the early balloonists was that they went up too rapidly and many of them passed out. And they had incredible problems at altitude, so that, for example, some of the early balloonists found that their arms and legs became paralysed and they couldn't actually pull the release cord to allow themselves to come down quickly. So why don't penguins' feet get frostbite when they stand around on the ice all day? Penguins actually allow warm blood to go to their feet, which keeps the temperature up. But of course they also lose heat from their feet. And that's why they have, that's actually quite useful since the rest of the body is really full of fat. So the only place that they can lose heat from is their feet. Whereas when I put my foot into the cold water like this, my feet get cold, and so the blood is directed away from my feet. It's channeled into the deeper vessels. So it's the middle of summer and I'm sitting outside in the late afternoon and it's freezing cold and I'm shivering. But Phoebe is not. Like other babies, she doesn't shiver. So the question is, how does she manage to keep warm when it's cold? Phoebe, like other babies, has little pads of special fat which run down her back here and up the back of the neck, which generate heat, rather like an electric blanket. Whereas the fat that you and I have acts more as an insulating blanket rather than an electric blanket. Come on, smile, smile. Yes. Well, you know what they say, babies always steal the show. What are the problems that the human body encounters when you go underwater? Sinking is always a problem, and breathing is the second one.
The air that I breathe that's in my tanks is the same as the air in the atmosphere. It's about 70% nitrogen and 21% oxygen. And this creates a problem because it limits the depth to which I can dive. Below about 30 metres, nitrogen acts as a narcotic. It's a little bit like alcohol. It makes you feel a little bit drunk. That can be a problem if I'm diving. And oxygen at pressure can also create problems. We don't really understand why oxygen should be toxic, and we have no idea about the high-pressure nervous syndrome that gives you the shakes. So there's still an enormous amount for us to discover about how the body reacts to pressure, which is a very good excuse for physiologists like me to go on diving expeditions.
All these questions concern the way the human body adjusts to a whole host of extreme situations. In the end, Fran's curiosity was consumed by something much more everyday.
Well, as I'm sitting here, my body is in balance. And one of the things which is held constant, which I am particularly interested in, is the level of sugar in my blood, which is normally held at a constant level, but is of course subject to all sorts of outside influences, such as if I was to eat one of these wonderful cakes, or if I was to go outside and run around taking exercise. Sugar is used by the cells in the body to generate energy. So when you eat a cream bun, for example, it's digested by the digestive system down to the basic constituents, which is glucose and sucrose. And those are used, glucose is used by all of the cells in your body to make energy.
Fran is interested in how this system breaks down. Insulin is the only hormone that can turn sugar into energy. In a diabetic, the problem is that insulin is no longer produced. With treatment, a diabetic can do very well indeed. Gary Mabbutt was captain of Tottenham Hotspur and he's played for England on 16 occasions. He's come to Trinity College, Oxford to talk to Fran about diabetes.
So, Gary, I wanted to ask you, when did you first get diabetes?
I was first diagnosed when I was 17 years of age. I was already a professional footballer with Bristol Rovers. And of course, as we know now, the classic symptoms of being a diabetic, the raging thirst, going to the toilet all the time. I was feeling very, very tired, very lethargic. And in the end, I was sent to the club doctor. And within a minute, he said, well, Gary, I'm afraid you're a diabetic. Now, to me at that stage, all I knew about diabetes was it was something to do with injections. That was all I knew. I hated injections. They always sort of had a fear of them. And of course, getting used to that, it took me a few days at least.
Could you tell us how you measure your blood sugar level? I mean, the cells which I work on measure the blood sugar all the time, but you have to do it for yourself. So could you just explain to us how you do that?
Yes, well, when I was first diagnosed, of course, then it was all urine testing. Now, of course, with technology and coming down a few years from then, now you can do it very quickly with a blood testing machine, which I just happen to have brought one with me. So it's my little bag I carry, you see, and it's got all my syringes and insulins. And of course my blood testing machine, which is very important. It's like a credit card size, that's all it is. Opens into a little pack and it's very easily done. In fact, I'll take you through the process. When I actually prick my finger in a minute, only I need to say ouch. You watching at home don't need to. If you want to, you can join me as well. But that's a simple machine, just click it there and you just put it on the side of your finger. Just click in there, that's all it is. Tiny little prick, and squeeze it to get the blood out. Get a nice drop of blood. And then you take the machine and you just put the blood onto the little mark at the end there and push the button. And then it counts down for 30 seconds and then it gives you your blood sugar reading. So I'll be doing this at, say, 5 minutes before kick-off and come in at half-time during a match, and as soon as the game finishes, I'll be doing these tests to find out where my blood sugars are. Hopefully it's where it should be, and everything's fine. If it's not, you may have to readjust, but hopefully it will be.
Wonderful, very quick and easy.
There are two forms of diabetes. Type 1, which Gary has, develops in people while they're still young, and type 2 can develop as you get older.
Fran, you're continuing your work into diabetes research. What exact research are you doing at the moment?
Well, I don't work on type 1 diabetes. I work on type 2 diabetes, which, as you know, is actually more common than type 1. And the difference between type 1 and type 2 diabetes is that whereas in type 1 diabetes, the cells in the pancreas that secrete insulin actually die, so that you have to have insulin for the rest of your life. In type 2 diabetes, that's not what happens. What happens is the cells still have insulin inside them. They just don't release it in response to glucose. So what we're trying to understand is why they no longer respond to glucose. And so the first thing that we need to understand is what happens in the normal cell under the normal conditions. How does glucose stimulate insulin secretion? And that's what we're particularly looking at.
In a healthy person, insulin is released by special cells in the pancreas known as beta cells. They can sense how much glucose is in the blood and release insulin when the level goes up. Fran wanted to find out how this system works.
We know that all cells have a membrane around them. And in the case of the pancreatic beta cell, it also has lots of small vesicles which are full of insulin. And they also have a membrane around them. And the key is that these vesicles must move to the surface membrane and make contact with the surface membrane. And then the two membranes fuse together, and then the contents of the insulin vesicle are released out into the bloodstream. So the question is, what causes that release? Because we know that when glucose goes up in the bloodstream, then insulin comes out. And there is one protein which plays a very important role in that process, which we discovered, which is known as an ion channel, an ION channel. And this is a particular type of protein that allows ions like sodium and potassium to move through it when it's open. So the channel that we are interested in is a potassium channel, and it sits in the membrane of the pancreatic beta cell. And at low glucose concentrations, this channel is open so that potassium ions can move out of the cell. Potassium ions move out of the cell. And when glucose levels go up, what happens is the cell breaks the glucose down to produce a substance which causes this channel to shut. And because this channel is shut, that triggers a chain of events that results in insulin being released. So the whole key to this process is when the channel is open, insulin is not released, and when the channel is shut, insulin is released.
What was your great discovery?
It's only a small discovery, I'm afraid, but what we were able to sort out was something about how the pancreatic beta cell releases insulin. So we identified this potassium channel in the cell membrane, which is regulated by glucose. And we showed how it is regulated by glucose. The big excitement was when I actually first saw glucose shut this channel. And that happened quite late one night when I was working all on my own. And, there was a lot of excitement, and there's a lot of excited swearing, actually, that goes on the tape when I found this out.
Fran had a lot to be excited about because she'd made a really big breakthrough. Together with her team of researchers, she discovered something about insulin release which had never been known before. These ion channels are studied with a special electrode, seen on the right, which is lowered onto the cell membrane to form a seal. The electrode records the tiny electric currents that flow through the ion channels as they open and close in the cell membrane.
Stefan, how's it going?
Well, it's going very well. I'm just putting in another electrode for the mutation I looked at earlier on.
Don't you get bored after years of looking at ion channels?
No, never. No, science is one of the most fascinating things there is. There's so many questions. Every question that you ask generates a new one. That's the excitement. And you are always doing different things. It's never the same.
As well as doing research in her lab, Fran also teaches at Trinity College.
Anybody can come to Oxford or Cambridge, providing that they have the intellectual abilities. It doesn't require that they've been to a public school, because, for example, I'm a case in point. I went to Cambridge from a tiny little village school originally. And the marvellous, marvellous thing about going to Cambridge was I discovered that the only thing that mattered was that you were interested in the work itself, in ideas. And I discovered, I think, that ideas and, well, your mind was far more important than the amount of money you had. And it was a complete revelation. And the other thing which was so important for me was that I discovered that it's okay to ask questions. And the questions that I asked and the ideas that I had were just as important as those of the senior academics.
So what about altitude? That's what we're supposed to be doing today, isn't it? So did you have any particular questions that you wanted to ask? Or...
Now Fran is at the top of her profession, but it hasn't all been plain sailing.
Pulmonary edema, yes. A lot of the problems that I have in my life is that I don't feel I'm good enough at anything. At anything, whether it's science or writing books or looking nice, whatever it is, I never feel that I do it well enough. But what keeps me going is just general interest. Maths and physics were never my strong points. But if you really want to do something, if you really want to know, all you have to do is to go out there and do it. And it doesn't matter at all where you come from or the sort of person that you are, you can do anything. And that's something I've found. And it's very important for women to believe that, because so often people are put off by thinking that this is not a job for a woman. And this is so untrue. You can do absolutely anything if you want to do it enough.